Human civilization travels through the plague. In this long human history, human beings often fail when facing infectious diseases until the emergence of vaccination. In the last 200 years, vaccination has played an irreplaceable role in reducing mortality and increasing the average life expectancy. The development of each new vaccine represents a great victory for humans in the fighting against infectious diseases. SINOVAC has never stopped to provide the vaccines to eliminate such diseases. While time advances, the challenge that human health is facing more and more complicated. When many diseases have not been completely defeated for many years, no matter it's SARS, avian influenza, hand-foot-mouth disease, or H1N1 influenza and COVID-19, the sudden appearance of these newly emerged diseases has sounded the alarm to human society again and again. While humans are fighting against diseases, they also witness the miracle when vaccines saves life. Every time, when facing uncertainty of a new disease outbreak and under great pressure, SINOVAC always stepped forward and quickly joined the front line of epidemic prevention. To contribute to the national as well as global disease prevention and control is the responsibility that SINOVAC takes. Every single dose of vaccine carries the great expectations for an individual, a family, or even the entire society. In the laboratory, SINOVAC's research and development team is rigorously and orderly carrying out the vaccine development every day to search for effective solutions to prevent and control infectious diseases. In the production plant, the production process in compliance to international standard and quality control system that covers the entire life cycle of a vaccine, guarantees for every dose of vaccine from SINOVAC, which deserves the trust of life. In the community, when SINOVAC's vaccine is timely delivered and safely provided to people, it will provide effective protection. Through the research, development, production, and application of high-quality vaccine, SINOVAC's vaccine brings hope of bringing a healthy life to people and ensuring harmonized development of the society. Today, SINOVAC's vaccines are providing effective protection against infectious diseases around the globe. When looking into the future, SINOVAC will make its best efforts to ensure the accessibility and affordability of human vaccines worldwide. SINOVAC aims to provide more and more high-quality products. SINOVAC allows more and more people to benefit from using vaccines. SINOVAC supply vaccines to eliminate human diseases.
Yeah, maybe we can start now.
Yeah.
Hello, everyone. So, Your Excellence, distinguished guests, ladies and gentlemen, good evening. On behalf of SINOVAC, I would like to welcome all of you to join this online conference. It is focused on the latest research and experience sharing on CoronaVac. My name is Xiao Lo. I work for SINOVAC Hong Kong as International Business Development Director for Asia Pacific. I know you are very excited to hear more from our speakers. So now we are very delighted to invite Mr. Meng Weining, and he's our Vice President for SINOVAC, to give an opening speech. Mr. Meng, please.
Thanks, Cheryl. Can you hear me?
Yes.
All right. Your Excellencies, distinguished guests, ladies and gentlemen, good evening. I'm Weining MENG from SINOVAC. It's my great honor to welcome all of you to be with us today and for international conference on the latest research and experience sharing on CoronaVac. Up to now, 17 November, up to November 17th, SINOVAC has supplied more than 2.2 billion doses of CoronaVac to more than 50 countries, including China, Indonesia, Malaysia, Chile, and Brazil, etc. More than 1.8 billion doses has been inoculated globally. There is a lot of experience in using CoronaVac globally to fight against the pandemic of COVID. In order to help decision makers to build better COVID-19 immunization strategies, SINOVAC and top scientists from China, Chile, Brazil, Indonesia, Turkey, and other countries has carried out numerous outstanding studies, including basic science, clinical studies, real-world studies, etc. These results prove that CoronaVac is safe and effective for different types of population in the world. Thanks to all scientists and partners, we move at such extraordinary speed while always maintaining the highest standard on quality and safety of our product and service. The name of event today is co-benefit, meaning go together with co-benefit. We aim to build the community of common health and mankind, because partnership and science are the key to bringing the world back to normal. We would like to build a platform for countries to exchange their experience of vaccination, because we can only rely on the joint effort from all of you to make such real change. Today, we invited the principal investigators of latest studies of CoronaVac to share their findings. We also invited national experts from different countries to share the immunization strategy and execution with COVID vaccines. We also invited scientists, experts, partners, and friends from over 50 countries, as well as international organizations together with SINOVAC Scientific Advisory Board member. SINOVAC leadership team is all participating in this conference today to witness this great achievement made on science and partnership. Thank you very much.
You have to
you're on mute.
unmute.
I would like to thank for Mr. Meng Weining for his warm welcome address to all the participants. Next, we are delighted to invite Secretary Carlito Galvez from the Philippines. Secretary Galvez is appointed as the Presidential Peace Advisor in 2018, and he is also the Chief Implementer of National Task Force Against COVID-19. And besides, his government vaccines are leader of National COVID-19 Vaccination Roadmap and Implementation Plan. Please join me to welcome Secretary Galvez.
Your Excellencies, Ms. Helen YANG, the General Manager of SINOVAC Biotech, Mr. Weining MENG, Your Excellencies, and other officials, staff, scientists from the SINOVAC Life Science, and to our fellow participants from all over the world, a good day to all of you. First of all, I commend the SINOVAC for organizing this conference. Gatherings such as these are crucial as they provide opportunities for stakeholders to touch base with each other and share best practices and jointly develop innovative solutions for the solutions of this pandemic. As we confront the difficulties caused by this health crisis, there is a greater need for us to collaborate, complement, and harmonize all our efforts in order to save humanity. This is the only way that we can adapt and thrive in the so-called new normal. The pandemic has truly tested the resolve of our nation. But what has enabled our countries to effectively address the challenges is to gather all the countries together to fight this common enemy and pandemic. Each of us had individual and collective key roles to play. For instance, in our country, while the national government takes leading a role in allocating deploying COVID-19 vaccine, the private sector and the medical community and other private organizations are regularly consulted to ensure that all our actions are aligned with each other. This is the reason why our national vaccination program is on track. As of November 18, our country has received a total of 129 million vaccines, more than half of which came from SINOVAC, and we thank SINOVAC for this steady supply that we have. And because of this, 72 million people have taken the jab, with 42.24% of the target populations are now fully vaccinated. We expect these numbers to rise significantly in the coming weeks as local government units continue to scale up their respective vaccination throughput. The latest data showed that the government has successfully created a balance between public health and economy. Our gross domestic product has expanded to 7.1% on the third quarter of this year, while the positivity rate of our COVID-19 infections is now below 5%. And our decisions to get the SINOVAC as early as February is really a good decision for all of us. This positive development are concrete proof that our country's pandemic response and mitigation efforts are bearing fruit. We are now seeing the light at the end of the tunnel as our nation's health situation continues to improve. SINOVAC CoronaVac vaccine has been instrumental in accelerating the implementation and expanding the coverage of our vaccination rollout. To recall, SINOVAC was the first manufacturer to deliver vaccines to the Philippines in February 2028 with the courtesy of our friend Chinese government. These life-saving vaccines enable us to begin the inoculation of our people in March, especially those who are highly vulnerable to the virus, such as the healthcare workers, senior citizens, and the people with comorbidities. These early deliveries were very critical as almost all Western-made brands at the time were in short supply as manufacturers could not be given the firm commitment on when they will be able to make the deliveries to our country. Indeed, SINOVAC, through Madam Helen, has shown and proven to be a highly reliable partner, as shown by its speed and consistency in delivering the vaccines steadily over the months, particularly at the last month of August, September, and October. In fact, SINOVAC has delivered the most vaccine doses to our country, which now comprises the main bulk of the supply inventory, more or less 54%. As of November 18, we have received 54.02 million SINOVAC vaccines doses, or more than 42% of the nation's total supply. SINOVAC vaccine has protected millions of Filipinos from the life-threatening effects of COVID-19 virus. The vaccine's capacity to provide a high level of protection, together with our excellent partnership with SINOVAC Life Science, means that CoronaVac will continue to be a major part of our country's national immunization program. On behalf of the Philippine government and the Filipino people, I would like to express our gratitude to SINOVAC Life Science, headed by Ms. Helen Yang and Sherry Law, and also Sir Weining MENG, that the Chinese government and Ambassador Wang Xilian for the assistance you have given to our country's vaccination rollout and for inviting me to this conference. We will always remember and we will not forget your act of kindness. And I would like also to commend all the attendees for joining this conference. Let us continue to work together to put an end on this pandemic as we move towards the road of economic recovery. Once again, a good day to everyone.
Thank you, Your Excellency.
So great thanks to Secretary Galvez to share with us the implementation plan against pandemic in Philippines. And next, I would like to introduce Mr. Datuk Zulkanain. So he is the him of Pharmaniaga Berhad as Group Managing Director last year. And he is also Adjunct Professor at University, Malaysia, Kelantan, and currently serves as the Chairman of UMK Business Venture and Fellow of International Institute of Plantation Management. Pharmaniaga is one of the Malaysian largest integrated pharmaceutical group. And early this year, Pharmaniaga is producing local fill and finish product, the coronavirus to supply in Malaysia. Please join me to welcome Mr. Datuk Zulkanain.
Ms. Cheryl Lau and distinguished speakers, Mr. Meng Weining, His Excellency Carlito Galvez Jr, His Excellency Budi Gunadi Sadikin, Ms. Helen YANG, Dr. Wang Xiangxi, and Dr. Liu Xiaoqiang. It is my pleasure to be at this meaningful international conference to share our momentous journey with SINOVAC COVID-19 vaccine and the national COVID-19 immunization program in Malaysia. When the COVID-19 has started to spread worldwide, we choose to work with SINOVAC Biotech Ltd on the fill and finish manufacturing and technology transfer to speed up the SINOVAC COVID-19 vaccine availability in the country. With their help, we have retrofitted our existing small volume injectable high-tech plant, Pharmaniaga Life Science or PLS, and train our highly professional technical team in SINOVAC COVID-19 vaccine fill and finish manufacturing. Initially, we have committed to supply 12 million doses of SINOVAC COVID-19 vaccine to the Malaysian government. However, we eventually supplied a total of 20.4 million doses to the government in 4.5 months ahead of schedule and more than 2 million doses to the private market. Our contribution to the national COVID-19 immunization program has significantly helped Malaysia to have now more than 95% of adult population to be fully vaccinated much earlier. Starting last month, Malaysia has given a conditional approval for a SINOVAC COVID-19 vaccine for adolescents aged between 12 to 17 years. And yesterday, the government has approved SINOVAC COVID-19 vaccine as a booster shot. And we hope SINOVAC COVID-19 vaccine will soon be available for children aged 3 to 11 years old. This shows the high confidence and trust that the Malaysian government has placed in Pharmaniaga and SINOVAC to continue deliver vaccines to protect the people from the pandemic. The success story of Pharmaniaga and SINOVAC partnership in Malaysia has garnered respect of the international communities, which has paved an opportunity for us to enter the international COVID-19 vaccine market. Last Saturday, we have started to deliver in stages 200,000 doses of PLS fill and finish manufactured SINOVAC COVID-19 vaccine to Myanmar. And we are exploring similar prospects in other Asian countries. Moving forward, we will continue to enhance our dedication to protecting the nation, which remains our priority, and we are forever grateful to be part of the history. I wish to put on record our sincere appreciation to our Prime Minister of Malaysia, Datuk Sri Ismail Sabri Yaakob, Minister of Health, YB Khairi Jamaluddin, Minister of Science, Technology and Innovation, Datuk Sri Adham Baba, Director General of Health, Tan Sri Noor Hisham Abdullah, Ambassador of the People, Republic of China to Malaysia, His Excellency Ouyang Yujing. These are among the key people supporting Pharmaniaga in this historic journey. My utmost thanks to SINOVAC for their trust in Pharmaniaga, and I wish our forged relationship will be stronger as we venture together in other areas. With that, I would like to quote a famous writer, Sir Winston Churchill. This is not the end. It is not even the beginning of the end, but it is perhaps the end of the beginning. Thank you for the invitation, and I hope and pray that all of us will one day meet face to face, shake hands, and smile to each other without the face mask. Thank you, and Xiangxi ZHANG, and good evening.
So thank you, Mr. Duka Sakana. So it is very touching to have your sharing. And we believe that the partnership between SINOVAC and Pharmaniaga, which indeed support a lot to the country and also the rest of the world, because you're talking about exportation. Thank you, Mr. Duka Sakana. Sorry. And next, we honorable invite Dr. Xiao Yiming to help us chairing the upcoming conference. Dr. Xiao is academician of American Academy of Microbial Sciences, and he's also chief AIDS expert of the Chinese Center for CDC. Let me give the floor to Dr. Xiao. Dr. Xiao, please.
Yes, thank you, Sherry. We would also like to thank all the other previously 3 speakers to share with us their experience in research to develop the vaccine, and through collaboration and cooperation to use the vaccine not only in China but in our neighboring countries. And we are very much like to thank all the efforts, the spirit of collaboration. This is also the spirit of co-benefit. Through hard working, through collaboration, through unite working together, we can co-benefit our people and our society, and also to end this fight against this once in 100 years pandemic. Without further ado, I would like to open the scientific exchange, and we know everything as to the good pandemic control has to be supported by good research. So in this session, we have both scientists and also our health officials to share their experience with us. I would first like to honorably invite the first speaker, His Excellency Budi Gunadi Sadikin, Minister of Health of Republic of Indonesia. And so Minister Sadikin, in addition to this health minister position, he's also a business people. He has been the board member of Indonesia Bankers and also for the international financial area positions. And his title for today is the latest tactic on Indonesia National COVID-19 immunization program and the best practice sharing. Your Excellency, Mr. Minister, the floor is yours, please.
Thank you, ladies and gentlemen. Good evening. First, I would like to thank SINOVAC because you delivered the first vaccine for Indonesian people. So on behalf of the Indonesian government and Indonesian people, thanks a lot for your contributions on those vaccines. And second, I just want to give you a brief development on Indonesian vaccination program. I joined as a Ministry of Health around 10 months ago. I have zero medical background as I graduate as a nuclear physicist and then 30 years in banking. So I don't really understand why the president chose me here, but I believe he would like to do it because he would like to see this vaccination is done as quickly as possible. So can I have my chart on the screen, please? What I would like to share with you is, next slide, quickly is, next slide. When the first time we designed our vaccination program by the end of 2020, the original design is for vaccinating 181.5 million Indonesians with the age over 18 years old. So we need around 363 million doses of vaccine. And I remember by the end of 2020, getting access to the vaccine were extremely difficult. So we are opening an early B2B discussions with several vaccine producers like Pfizer, AstraZeneca, Moderna, SINOVAC, Sinopharm, CanSino, and several others. But yes, to be honest, the quickest one was SINOVAC. SINOVAC dealt with Biopharma to secure the first millions of doses of vaccine. And those doses of vaccine arrived by the end of December. So we started our vaccination program on the 13th of January. In the middle, I believe around July or June or July, then the vaccine usage can be extended down to adult or young children with the age of 11 to 18. So that is where we can, then we add additional target from 181.5 million to become 208 million people. So we need additional around 50 million doses, and it comes to 416 million doses. We, as the other countries, we schedule the vaccination based on risk. So the healthcare providers is the first one to receive the vaccine, followed by elderlies and public officers, and then as what we are doing now is a general public and also young adult between 12 to 18 years old. And the pictures there were our president when he was vaccinated on the 13th of January 2021. Next slide. Now, we had a very slow start. Not because the capacity of the infrastructure, but more because of the number of vaccine that we can access on the beginning of this year. But after August, then we can ramp up our vaccination activities because we have enough vaccine that we can secure starting on August. Now, Indonesian position is number 5 for number of people who receive at least the first dose. And we are also number 5 in terms of number of vaccine doses administered after China, India, United States, and Brazil. Next slide. So when we what this chart I would like to share with you is again the speed. So our first 50 million doses we reached around 26 weeks. So we started on the 13th of January. We reached our 50 million doses on the 8th of July. And then the next 50 million doses we reached in 7 weeks. So 8th for July until 31st of August. And then for the next 50 million is another 5.5 weeks. And then the last 50 million, we can do it exactly 1 month from 6th October until 4 November. Again, it depends really on the availability of the vaccine. As of yesterday, we have vaccinated 219 million doses, 232 million Indonesian for first dose, and 86 million Indonesian 2 doses. So we have vaccinated 63% of our target population because Indonesia $208
Yeah, the connection is not good. Maybe the line speed.
I lost the connection for a little bit.
Okay.
So I believe some of the virus has got a breakthrough infection and enter my internet network. So we have to, maybe it is a new epsilon variant that we haven't found recently. But and anyway, so Kiki, can you move to the next slide, Kiki? Next slide. So as you can see, this is the progress. Now I can share with you the dynamics beyond this data, which is what interesting. When we start early, I remember we only do 100,000 jabs per day. And many people, including the famous magazine, The Economist, mentioned that if Indonesia do this, then Indonesia will finish the vaccination in 10 years. I didn't get the chance because at that time we are extremely busy to explain to The Economist, but I just got the time just this morning. And I talked to them, the reason why I only do, 100,000 jabs per day, because I only have 3 million doses available from SINOVAC. So if you remember at the beginning, SINOVAC sent us the ready-to-use vaccine. It's not a bulk vaccine. And after that, you send the bulk vaccine, but bulk vaccine, it takes time, to produce, to become ready-to-use vaccine. So I only received the first 3 million. And I said, 3 million, if I divide by 30 days, so I can only do 100,000 a day. I cannot do more. If I do like 1 million jabs per day, and suddenly after 3 days, all Indonesians will scream to me and asking for vaccine. So we intentionally pace the speed of the vaccination program. After July, when we receive enough vaccine, then we can do 1 million jabs per day, because we have between 30 to 50 million doses of vaccine every month. And then when we have more vaccine, we move it higher. Our record is 2.4 million jabs per day. And for the last 5 weeks, 6 weeks, we actually always reach close to 2 million jabs per day, which is, if I compare with other countries, I just had a meeting with my colleague G20 in Rome. I think we are the highest, even compared with Western country like United Kingdom or even United States. Recently, we did faster than them. But of course, we are behind China and India. You are at a different league. You are at a different league. But what I can share with you, on March, I realized that it will be difficult to get the tractions. Why? Because our system, logistics system, we send the vaccine to the provinces, and then the provinces will distribute the vaccine to the cities. That is the system used by the previous government before the crisis in 1998, where everything is centralized. But with the reform of Indonesian political system, after Suharto went on, after our famous president, then we decentralized the government. So the issue is that the local government is being elected. So they're not really 100% the employees of the central regime, because they're elected by the people. Then it created issues, because they are also political appointees. And then when we distribute to them, it's not 100% equally distributed to all the cities that have a different political affiliations. So we noticed that, and I think it happens also with other countries that have a similar political system. Then we start sending directly to the cities and regencies. I believe it started on March. So there is a certain surge on March about that. But then after that, you can see a drop. It is because then we realize that the regencies and cities across Indonesia, they don't have the similar capacities in do vaccination program. So what we did, the end of April, we involved the private sector, because the private sector want to get vaccinated as quickly as possible. So after 30 years as a banker, I have access to all of them. So I called all these rich families and big corporations, and I said, why don't you help me vaccinate the people? We will provide you the vaccine, and you create the vaccination centers in the cities that you operate in. So when you saw there is an increase in May and early June, that is because of that initiative. But then we realized that when the vaccine, the ready-to-use vaccine will come in a large amount, we measured that the capacity to do the vaccination will not be enough using this channel. Then by early of June, I talked to the president and asking the president to assign the army and police to help us. Because Indonesian army and Indonesian police are controlled by the central government. They have offices everywhere in our 13,000 islands, 514 cities, and they are very, very disciplined. And they have doctors and nurses also with them. So when you start seeing the significant increase in July, it's because of the army and police are helping us. And then when on August, we have enough doses of vaccine, as you can see, then we have the momentum already. So we have the momentum already. And now we reach around 2 million doses. You might notice that in the last 2.5 weeks or 3 weeks by now, it's declining. It's very funny, actually, and it is related with SINOVAC also. And when the first time we start with SINOVAC because it was the only vaccine that we have. A lot of people are challenging in us, including our own people in Indonesia. Why did you choose SINOVAC? Because the efficacy is low. Why don't you take Pfizer or Moderna or AstraZeneca? And I said the only reason is because SINOVAC provide the vaccine the fastest to Indonesia. And based on the discussion with all the epidemiologists, the fastest you can vaccinate your people, the better you are. So we just want to do it quickly. And that creates an excellent branding for SINOVAC in Indonesian people. Especially SINOVAC effect after vaccination is considered very, very mild. Very, very mild. So when we start having another type of vaccine, AstraZeneca, Pfizer, Moderna, where the after vaccination impact is actually, more severe than SINOVAC, so people get afraid. Now what happened in the last 3 weeks is SINOVAC has come to the peak, the order, because now the new batch of AstraZeneca, Pfizer, and Novavax start coming into Indonesia. So SINOVAC has been used, I think close to 100 million or more than 100 million doses. And then we start vaccinating using Pfizer and AstraZeneca. And to my surprise, this is again the emotion of the people. They're still looking for SINOVAC because they believe that, oh, it is more mild. You don't get hang out after vaccination. So then I had to re-educate the people again that, hey, vaccination, you don't care about the brand. As long as you get vaccinated, that is the best way to go. So now we are still struggling a little bit because in the last 3 weeks, when the volume of SINOVAC vaccination is being replaced by AstraZeneca and Pfizer, the public perception in Indonesia, which is good for SINOVAC, is still very SINOVAC minded, very SINOVAC minded. Next slide. So this is the schedule of the vaccine delivery. Actually, the original schedule, we can get 460 million doses. So enough for vaccinating 208 million. But starting on October, the end of October, we realized because we have a lot of vaccine coming in on September, and especially vaccine that come from COVAX, Gavi, then they have a very short expiry date. When expiry date is very short, sometimes it's very difficult to distribute to the remote area because from January to August, we already vaccinating the large cities. The easy part, the large city, large population, we are vaccinating them. So starting on September, we start to go to small cities, which is logistics is more difficult, transportation is more difficult. And then during the time, we have a lot of vaccine coming in, and including vaccine on September coming a lot from COVAX, Gavi. This is a free vaccine. Actually, they want to give us 108 million this year. But when I see the capacity of our systems and also that the short expiry date, let's delay a little bit. Because now we still have around 50 million vaccines stocked all over across the cities and provinces in Indonesia. So we postponed about 50 million to 2032, first quarter, which is a month period, usually 50 million doses. And we will end up around 418 million doses. And as you can see, we have many brand vaccination. We have SINOVAC. I think SINOVAC is the most, and then followed by we have AstraZeneca, we have Pfizer, and we have bilateral grant from several cities, 22 million already. It is a free vaccine from our neighbors. And mostly Pfizer or AstraZeneca with some Moderna, and also we will receive we are receiving 60 million doses from COVAX Gavi out of 108 million committed doses from COVAX Gavi. But COVAX gave us quite a lot, because they see that we can do the vaccination quickly. We can do the vaccination quickly. Next slide. So our target is Indonesia is very spread, the country. Infrastructure is also different among cities. And our target is for every provinces, 34 provinces in Indonesia, our target is to achieve 60% of the target population's first dose and 70% of the target populations by December. So 60% first dose by November, 70% first dose by December. Usually when we reach the first dose, it's between 4 to 6 weeks that it will complete the second dose. It will complete the second dose. And we have seen that several countries are lagging, especially Papua, you know, our eastern part island, and then Aceh. Aceh is also the western side of Indonesia. So our job now is send our resources, the armies, the police to accelerate the vaccinations in Papua and Aceh. So that is where the number of vaccinations is the lowest. Next slide. I think the key message that I would like to share with you is it is something, the vaccination program at this scale is unprecedented for Indonesia. And I think I also believe for many countries. Usually we only do like 30 million to 50 million vaccinations a year. Now we have to do close to 400 million vaccinations per year. It is like 8 times the normal volume that we do. And our geography also make it much more difficult, the situations, because we have 13,000 islands and it's separated by sea. So it's make it more complicated. So since the beginning, I believe that it is not something that we can do individually. This is a program that we have to do it together. It is something that the government cannot, you know, exclusively push this program, but we need to inclusively involve private sectors, the regional government, you know, the police and army to make sure that we can execute this vaccination program as quickly as possible. And I'm happy to see the result is because now every Indonesian believes it is important and they want to do the vaccination program either at the small scale, your community level, or your alumni of one university or your alumni high school, and they do it on their own expenses. So the final things that I would like to say is we can do this because now the Indonesians see this vaccination not as the government program, but as a national movement. In program, somebody owned the program. In movement, everybody owned that program. So everybody believe that we need to be vaccinated and we need to work together to get our communities and families vaccinated. Indonesia is Indonesian culture is very social. The social capital is extremely large. As long as we can consolidate the social capital and unleash the social capital or our people, actually that can really be so powerful and help the task of the government. So that is a short story of the Indonesian vaccination program. Again, I want to thank SINOVAC because you are the fastest and you are the first one to deliver the vaccine to Indonesia. So on the behalf of the Indonesian government and Indonesian people, please accept my gratitude. What you have done is not only saving Indonesian lives, but it's also a tribute to humanity. Thank you.
Thank you very much, Your Excellency, Minister Sadikin. And your sharing the experience is so novel and so exciting. I could find that co-benefit is really reach the most optimal resource. I could see you in your leadership with your president's support, you can change the decentralized distribution system to direct send vaccines to the cities. And also with your background as a financial businessman, you utilize the private sector to help doing the vaccination centers. And also the third innovation I could see, you use your army and the policemen to help to distribute the vaccine and in the over 30,000 islands with the speed of the number 3 in the world, faster than the American, European countries. It's a great achievement. We could also understand your new challenges with the SINOVAC vaccine. Even people are like that, low side effect, but they are maybe a little bit worried about his the efficacy. I think our next speaker will show us data for the booster shot. After the booster shot, the SINOVAC can be as good as like a Pfizer, AstraZeneca. So this is my pleasure to introduce Professor Xiangxi WANG. He's from the Chinese Academy of Sciences, and he will tell us today about the third dose of inactivated vaccine. It means SINOVAC. To augment the potency, breadth, duration of the vaccine response. The floor is yours, Professor Wang.
Thanks, Professor, for introduction, and thanks the committee for invitation. Firstly, I share my slides. Okay. It's my great honor to be here to report some progress on the third booster administration of an inactivated vaccine, CONVAC. I'm Xiangxi from the Institute of Biophysics, Chinese Academic of Science. Prevention and control of infectious diseases are strategy requirement of global public health. I am a structure biologist. Actually, virus structure information can facilitate the research and development of virus detection, vaccine, and drug design. In the past 5 years, my lab used cryo-EM to determine the tense of virus structures, including the recent SARS-CoV-2 and some giant virus like herpes virus and African swine fever virus. I'm always impressed by
you need to share your slides.
Yeah, I shared.
We can't see.
Yeah, we can't see it.
Can't see it. Okay, let's do it again.
Yes.
Yes. Okay.
Yeah.
Okay.
Maybe you put it on the, yeah, enlarge it.
Okay.
Thank you.
Okay. I'm always impressed by the amazing beauty of the virus structures. Let's see the very beautiful structure of virus structures. They look very beautiful and like artwork. As we surface proteins like spike in SARS-CoV-2 has two distinct conformations, pre-fusion and post-pre-fusion and post-fusion conformation. In their pre-fusion conformation, most neutralizing epitopes are exposed, and the antigen with the pre-fusion conformation can always elicit high levels of neutralizing antibodies, being the effective components for vaccine. However, the post-fusion so a totally different role in the antigenicity. So a better vaccine should contain more perfusion antigens. For SARS-CoV-2 spikes, as we expect, there are also two conformations, pre-fusion and post-fusion, two conformations. Furthermore, SARS-CoV-2 spikes are very fragile and can easily be stripped off the variants, as seen in these two figures. When we firstly checked the sample under the negative stain EM, most spikes have been stripped during the purification in the left figure. After the optimization in purification procedure, a large abundance of spikes are decorated on the variant and adopt a perfusion conformation. So that might be the one of the reasons for the CoronaVac. A dose of 3 microgram of inactivated virus rather than 50 microgram for the Moderna vaccines can elicit reasonable neutralizing antibody titers in animal models and human beings. So inactivated virus vaccines have exhibit a very promising protective efficacy in clinical phase and real world. The ongoing COVID-19 pandemic has lasted for nearly 2 years. Several types of COVID-19 vaccines have been deployed at a large scale, but progress in halting this pandemic seems very slow due to possible reasons. One is that the emergence of variants of concern, VOC, such as the alpha, beta, gamma, and delta, and lambda. These VOCs appear to be highly transmissible and more resistant to neutralizing antibodies. Because these VOCs contain multiple mutations in their spike. Some of residues are the key epitopes located within the epitopes of some neutralizing antibodies. The other reason is that the neutralizing antibodies wane over the time, either after vaccination or the natural infection. Approximately 80 to 90% of antibody titers waned 6 months after the second vaccination, even including the mRNA vaccination. So a plausible approach to solving this problem is the administration of a third dose of the vaccine, somewhere between 6 and 12 months after the second dose of vaccination, for enhancing and prolonging the protection. serologic analysis of plasma samples from early convalescence, people received 2 or 3 doses of inactivated vaccines. So a third dose booster of inactivated vaccines lead to the enhanced neutralizing breath to the SARS-CoV-2 variant. That means a third dose immunization not only boosts higher neutralizing titers, but also more resistant to VOCs compared to 2-dose vaccination. Similar to MR vaccination, over 80% of neutralizing titers decreased 6 months after the second vaccination, while the third dose administration of SINOVAC boosted these titers by 20-fold at 2 weeks post-vaccination. Notably, neutralizing antibody titer was maintained at around 60 at 6 months post the third dose booster. This high titer is comparable to the high level of neutralizing titer elicited by the 2-dose administration. To unveil the underlying mechanism, we then systematically analyze the SARS-CoV-2 specific B-cell repertoires from the early convalescence, 2-dose or 3-dose vaccines. The number of nucleotide mutation in the V gene in 3-dose vaccines is higher than those in both 2-dose vaccines and early convalescence. This indicates ongoing somatic hypermutation of antibody genes and mutation for antibody binding affinity. In line with this, antibody isolated from 3-dose vaccines, so higher binding and neutralizing activities, as well neutralizing breath to most VOCs. Subsequently, we analyzed a total of nearly 200 available structures of SARS-CoV-2 in complex with their neutralizing antibodies to decipher the structure activity correlates. Unveiling ultra potent VOC resistant and broad spectrum antigenic patch. Overall, RBD targeting neutralizing antibodies can be divided into 6 classes, and NTD targeting antibodies are classified into the 4 types according to their binding size. The first 3 types of antibodies target the RBM receptor binding motif, involving extensive interactions with residues such as K417, E484, L452, and residue and N501, and so on. And so significant decrease and even complete loss of activity against the VOCs. But the last 3 types of antibodies recognize relative conserved epitopes and show a broad neutralizing activity against most VOCs, and even against pan-sabicovirus. With a deep understanding of antibody types, we then investigated the frequency of distribution of 6 types of neutralizing antibodies in early and late time point. The early time point means analysis from the early convalescence and 2-dose vaccines. The late time point indicated the analysis from the later convalescence, all the 3-dose vaccines. The first 3 types of antibodies were dominantly existed in the early time point, explaining the reduced activity against VOC. But in late time point, the last 3 types of antibodies have significantly been enriched, almost equal to the first 3 types of antibodies. This evidence provided evidence for observed increased neutralizing breadth in 3-dose vaccines. In summary, a 3rd dose of inactivated vaccines lead to expeditious and durable recall response that is more potent with significant enhanced neutralizing breadth. The ongoing antibody somatic mutation and the memory B cell clonal turnover via 3-dose inactivated antigen stimulation confer to the neutralizing potency and breadth. A total of nearly 200 available complex structures of SARS-CoV-2 encompass their neutralizing antibodies. Deciphered the structure activity correlates, unveiling ultra component VOC resistant and a broad spectrum antigenic patch on the RBD. Differential frequency of distribution of antibody types from the 1 to the 6 contributed to the observed enhanced neutralizing breadth of the plasma in the late convalescent and 3-dose vaccines. The measured binding affinity vs the SHM rates indicates the binding affinities increase accompanied by the SHM mutation increase. Lastly, I would like to thank all my team members and all my collaborators. Thank you for all, and thank you for listening.
Thank you, Dr. Wang, for your excellent report. We are very happy to see with the 3rd post injection, the inactivated vaccine can be very strong in generating neutralizing antibody, and the titer can last more than 6 months. That is very good news. And also thank you for your in-depth analysis for the mechanism of those antibodies, including the broad neutralizing ones. That is very encouraging. Could you stop sharing your slide?
Okay. Yeah.
Yeah. So this is very good compared with HIV-1. HIV, broad neutralizing antibody, need a much longer time to develop. So therefore, COVID-19, the SARS-2 virus, we can quickly use vaccine and generating good protective antibodies. You have not terminated your slide sharing. Stop.
Stop. I have stopped.
我们这边可以控制吗？ we still see your last slides on the screen. Oh, now it's good. Yeah. Yes, it's my pleasure to introduce the next speaker. We all know that in addition to efficacy, the equally important is safety. The next speaker is from the Yunnan Center for Disease Control and Prevention, Dr. Liu, Xiaoqiang LIU. And Dr. Liu is the director of the Vaccine Clinical Research Center of the Yunnan CDC. He will report to us about a review on the safety data from the inactivated vaccine COVID-19 vaccination. Thank you. Dr. Liu, the floor is yours.
Thank you, Dr. Zhang. Thank you for your introduction. And okay. Can you see my slides?
Yes.
Thank you very much. Dear Chairman and Professors, I'm Liu Xiaoqiang from Yunnan Provincial CDC, and my presentation is about the interim result of safety observation on the inactivated SARS coronavirus disease 2 vaccine. It's viral cells in population aged 18 years and older. The study was sponsored by SINOVAC. On 5th February 2021, SINOVAC obtained the conditional approval. There are two conditions needed to be verified. The first one is to verify the condition of the safety observation in large-scale use, including in population with underlying medical conditions. And the second one is to discover the possible AES and the SAE. The objectives of this study are to evaluate the safety of CoronaVax among a population aged 18 years and older in a large-scale use, including people with underlying medical condition, and to provide evidence to immunization strategy of the COVID-19 vaccines. We conducted this study in 31 counties from the 11 provinces, which located as the north, south, eastern, and western of China. A total of 121,000 subjects were supposed to be recruited. Each province needed to recruit at least 11,000 participants, with no less than 2,000 people suffering from the underlying medical conditions, and no less than 4,000 people aged over 60 years, respectively. The inclusion and the exclusion criteria are the same as the instruction of the vaccine. And also, all subjects need to sign the informed consent. We started the indicators of safety observation, including the associated local and systematic AE, other kinds of the AE such as the unusual or real AE, and we also correct the adverse events of the special interest, such as the bell palsy and sudden diabetes, hearing loss, and also the GBS. And we focus on the SAE also. In this study, the primary parameters is the incidence of the AE within 7 days after each dose of vaccination in all participants. And the secondary parameter is the incidence of the AE within 7 days after each dose in participants with the UMC. We also focus on the incidence of AE within 28 days after each dose of the vaccination, both in all participants and the UMC. And also we have some exploratory parameters. We follow the procedural recruitment and informed consent, inclusion and exclusion criteria, and the first dose vaccination, safety observation, second dose vaccination, and safety observation. Whole follow-up will be finished within 1 year after the second dose of the vaccination. We collect the safety information both by e-collection technology and also paper-based daily record. Study was started in the May this year. Now, all sites completed the recruitment already. This result is the interim data analysis. Totally, we recruit about 118,000 persons, and 115,000 were eligible for the participation. Among the participants, 73,000 were 18 to 59 years group, and another is the over 60 years. In both groups, there are over 13,000 persons with UMC. In this analysis, information collected by paper questionnaire has not been imported into the designed system yet. So this is not the final result. This graph shows the distribution of the participants grouped by age and health situation. In our study, male participants account for 52%, while the female accounts for 47%. Han ethnicity accounts for the most rate is about 83%, while the other ethnicities account for 16%, with Tujia, Hui, and the E ethnicity. The main kinds of the underlying medicine condition or hypertension, diabetes, chronic gastritis. The incidence of the AE was 2.52%. Is lower level of the AE incidence. According to the age, the incidence of the AEs in adult and the elders group were 3.19% and 1.34% respectively, with the p-value is 0. According to the health condition, the incidence of the AE in healthy participants and those with UMC were 2.47% and 2.64% respectively, with the p-value of 0.1. There is no statistic difference between the health group and the UMC group. Most local AE or pain with the incidence 2.11% in adult and the 0.76% in adults. And the most symptomatic AE or fatigue and the headache, muscle pain, or AEs with no occurrence rate. This slide shows the incidence of the AE according to the healthy condition. No statistical difference were found between those two group participants. It's nearly the same occurrence rate. In this slide, we also found that for most AEs, adults always with no incidence. Among the different age and the different health condition, the same figures is that most of AE are grade 1. Only a few AE are grade 2. There's only a few in grade 3 and 4. It's nearly 1 or 2 cases. The incidence of AE after first dose of vaccination in adults and adults group were 2.40% and 2.76% respectively. After the second dose, the incidence of the AE in adults and the adults group were 1.42% and 0.78% respectively. According to the health condition, the incidence of the AE after the first dose of vaccination in healthy and the UMC group were 1.82% and 1.76% respectively, with a p-value is about 0.5. I say there is no difference. The incidence of the AEs after the second dose of vaccination in healthy and the UMC group were 1.13% and 0.35% respectively. This SE during serious adverse events during the observation period is death case. There is a death case, but our expert community confirmed as no adverse reaction, and there are also 2 other cases of the unsolicited AE that were successful to be ASI. The diagnosis are facial edema and numbness of the arms. So far, our interim conclusion is corona vaccine showed good safety in a large school population aged 18 years old and above, including those with underlying medical condition. The incidence of the AE or 2.52% with most events or solicited local AE and grade 1. And also the incidence of the AEs in the adults and the adults group were 3.19% and 1.34% respectively, with the p-value is about 0. With the healthy and the UMC group were 2.47% and 2.64%, respectively, and with the p-value is 0.1. We will continue the follow-up, and the interim results are subject to change, depends on the final result. This is my presentation. Thank you, everybody.
Thank you, Dr. Liu, for a very comprehensive safety evaluation. And that's really confirmed that the inactivated vaccine has a very low side effects. It's our colleagues, officials from Indonesia, who they found the similar results. I think the safety, because of the large-scale usage of the COVID vaccine, the safety is the top priority for all government and international organizations. So I think the collaboration, we have a different kind of vaccine. That is a very good thing for the world. Different vaccine has different technology, different efficacy and safety profile. Good use of and even combined use of them can meet the requirement for all different people with different conditions within a country and between countries. So that's a real, very important data, a very important results. We will have a short break according to the schedule. So that is if we have a 10 minutes break, we could come back at 8:30, maybe less than 10 minutes, 7 minutes break. So we will come back at 8:30. See you a moment later. Thank you.
Indonesia telah diberkati dengan kekayaan dan keindahan alam. Dengan berjuta pesona, kita dimanjakan dengan tempat-tempat terindah di dunia. Menikmati momen-momen membahagiakan yang membawa kehangatan. Dengan masyarakat yang kuat, kita pun terus berkembang dan bergerak maju. Namun, masyarakat terkuat pun harus diuji melewati masa yang sulit. Ketika dihadapkan dengan keadaan yang berat, kita pun dituntut untuk kembali bersatu padu dan saling menopang. Hingga akhirnya kita dapat kembali bangkit bersama. Melalui tantangan-tantangan yang ada, kita menemukan dan mengingat kembali arti kerjasama yang sesungguhnya. Permasalahan yang kita lewati menguatkan kita sebagai sebuah bangsa. Di masa pandemi ini, dunia pun melahirkan pahlawan-pahlawan baru. Mereka yang bertahan untuk tidak bertemu keluarga. Mereka yang bekerja tanpa kenal lelah. Mereka yang berkorban demi menyelamatkan banyak raga. Di hari-hari penuh kekhawatiran, kita pun menemukan secercah harapan. Kini vaksin hadir dan melindungi para pahlawan dan juga orang-orang yang kita cintai. Perlahan tapi pasti, melalui pengorbanan mereka dan dukungan dari kita semua, Indonesia akan kembali bangkit dan menyongsong kehidupan yang baru. Terima kasih atas perjuangan kalian. Selamat Hari Pahlawan untuk para pahlawan Indonesia.
Hello, good evening. And we are coming back. And we will have 3 speakers before we conclude. including Chinese and Chile colleagues. And the next speaker is Dr. Li, Minjie Li, from Hebei Center for Disease Control and Prevention. And he will report. So he's attending doctor at the Vaccine Clinical Institute, Hebei Center for Disease Control and Prevention. He will also report the safety and immunogenicity of the SINOVAC in 3 to 17 years old children in a phase 1 and 2 immune persistence clinical trial. Dr. Li, please, the floor is yours.
Thank you, Doctor. Good evening. Okay, yes.
Please share your slides, your screen.
Okay.
Yeah.
Okay, I'm Minjie, come from Hebei. My topic is safety and immunogenicity of current work in children aged 3 to 17 years in phase 1 and 2 clinical trial. I will report from the following aspects: practical introduction, study team, sponsor, SINOVAC Life Sciences, study institution, Hebei provides Center for Disease Control and Prevention. Set of the clinical trial, Center for Disease Control and Prevention of Zhanghang County. Objective: to evaluate the safety and immunogenicity of inactivated COVID-19 vaccine in population aged 3 to 17 years. Phase 1 clinical trial to evaluate the safety, tolerance, and preliminary immunogenicity of different dosage vaccine in health population aged 3 to 17 years. Phase 2 clinical trial to evaluate the immunogenicity of the safety of different dosage vaccine in health population aged 3 to 17 years, and to determine the appropriate dosage for further clinical evaluation. The study design is a single center randomized, double-blind, placebo-controlled phase 1 and 2 clinical trial. Study population: children and adolescents aged 3 to 17 years. Sample size: total of 572 subjects, with 72 in the phase 1 and 480 in the phase 2. Dosage: low dosage is 300 SU, and the medium dosage is 600 SU. Design is a phase 1 clinical trial. 72 subjects were enrolled in the phase 1 clinical trial, divided into 3 age groups. Each age group was divided into low dosage and the medium dosage. The groups were sequentially enrolled according to the principle of age de-escalation and dose escalation. In each dose group, 9 subjects were randomly assigned to receive the vaccine, and 3 subjects to receive the placebo. After 7 days of follow-up for safety were required before entering the next stage. Subjects receive 2 doses of vaccine on day 0, 28. blood and urine samples were taken before and on day 3 after each dose, and tested to investigate any abnormal changes of hematology, biochemistry, and urine routine indexes. Blood samples were collected on day 0, 20H, 56, 200H, 380H from subjects to evaluate the neutralizing antibody titers. Subjects were observed in the study site for at least 3 minutes after vaccination. The listed adverse events were recorded for 7 days after each dose and isolated adverse events for 28 days. The serial adverse events are recorded throughout the study, and the follow-up will continue until 12 months after the last dose vaccine. The phase 2 clinical trial used the same safety observation method. In the phase 2 clinical trial, a total of 480 subjects were recruited including 180 aged 12 to 70 years, 180 aged 6 to 11 years, and 120 aged 3 to 5 years. Subjects were randomly assigned to either low dosage vaccine, medium dosage vaccine, or placebo. Subjects were enrolled and received 2 doses of vaccine on day 0, 20H, and collected blood samples on day 0, 56. 240 subjects were collected blood sample on 3 months after the second dose, and another 240 subjects were collected blood sample on 6 months after the second dose. The new protocol planned to add booster immunization. Only subjects in vaccine groups received booster dose at 10 months or 12 months after the second dose. Implementing status. This is a clinical trial timeline. The phase 1 clinical trial was launched in October 31st, 2020. The phase 2 clinical trial was launched in December 12, 2020, and completed the second dose of vaccination in January 2021. There are some pictures of on-site investigator training, on-site assessment, process simulation, take-off meeting and training, informed consent, blood collection, vaccination. In the phase 1 clinical trial, 110 individual sent the informed consent. 38 were included. 72 were enrolled and completed blood collection before the first dose. 71 received the first dose. Before the second dose, at 28 days after the second dose and at 6 months after the second dose, 69 subjects completed blood collection. In the phase 2 clinical trial, 515 individuals were screened. 35 were included. 480 were enrolled and completed blood collection before the first dose. 479 received the first dose. 467 subjects received the second dose. 462 subjects completed blood collection at 28 days after the second dose. 227 subjects completed blood collection at 3 months after the second dose. And 233 subjects completed the blood collection at 6 months after the second dose. Safety evaluation is a phase 1 and 2 clinical trial. A total of 550 subjects received at least 1 dose of vaccine or placebo. The overall incidence of adverse events were 39%. The overall incidence of adverse events related to vaccination were 26%. The overall incidence of the adverse event related to vaccination was 20%. Adverse reaction. The overall incidence of adverse reaction was 26%. The overall incidence of local adverse reaction and systemic reaction were 14% and 15%. The overall incidence of solicited adverse reaction was 24%. The overall incidence of unsolicited adverse reaction was 6%. The most common adverse reaction were injection site pain and fever. Except for a higher prevalence of injection site pain into vaccine groups than that in placebo group, there were no significant statistic difference in the prevalence of other solicited or unsolicited adverse reaction among 3 groups. Adverse reaction severity. Most adverse reactions were grade 1 and grade 2 in severity. The incidence of grade 1, grade 2, and grade 3 adverse reactions were 20%, 11%, and 0.36% respectively. Only 2 subjects reported grade 3 adverse reaction, both fever and recovery. Immunogenicity evaluation. In the phase 1 clinical trial, 20 days after the second dose, the seroconversion rate in low dosage group, medium dosage group, and placebo group were 100%, 100% and 0% respectively. The GMT were 55, 117, and 2 respectively. Compared with 20 days after the second dose, the seroconversion rate of GMT decreased significantly in low dosage group 6 months after the second dose. In the medium dosage group, the seroconversion rates were still 100%, while the GMT decreased significantly. In phase 2 clinical trial, 28 days after the second dose, the seroconversion rate of neutralizing antibody in low dosage group, medium dosage group, and placebo group were 96%, 100%, and 0%, respectively. The GMT were 86, 142, and 2, respectively. Compared with 28 days after the second dose, the seroconversion rate and the GMT did not decrease significantly 3 months after the second dose. The seroconversion rate and the GMT decreased significantly 6 months after the second dose. In the medium dosage group, the seroconversion rate decreased slightly, while the GMT decreased significantly. In different age subgroups, the seroconversion rate 28 days after the second dose reached more than 92% after receiving the vaccine in all age groups. The seroconversion rate 3 months after the second dose exceeded 97% after receiving the vaccine in all age groups. 6 months after the second dose, the seroconversion rate in the low dosage vaccine and the medium dosage vaccine were 100% in 3 to 5 years old age groups, 94% and 100% in 6 to 11 years old age groups, 69% and 88% in 12 to 17 years old groups, and which were significantly decreased. The seroconversion rate of medium dosage group were higher than low dose group in age subgroups. In different age subgroups, the GMT 3 months after the second dose of vaccination, the GMT of subjects in all age groups had no significant downward trend, especially the GMT of children aged 3 to 5 years after the second dose was comparable to that of 28 days after the second dose. 6 months after the second dose of vaccination, the GMT showed significant downward trends in all age subgroups over time. Conclusion: favorable safety of CORONAVAC in population aged 3 to 70 years were confirmed. The incidence of adverse reaction showed no statistically significant difference among 3 groups. It was shown good immunogenicity in healthy population aged 3 to 70 years. The seroconversion rate and the GMT of medium dosage group were higher than that of low dosage group. There were no significant decrease of the seroconversion rate and the GMT 3 months after the second dose, while the GMT showed significant decrease 6 months after the second dose. It is showed that the population aged 3 to 17 years should receive the booster dose of vaccination. Thank you.
Thank you, Dr. Li, for another very comprehensive report on the safety of the inactivated vaccine. And this is, China is a country who started the 3 to 5 years old children vaccination. I think the safety data is very, very important, as well as the immunogenicity data. So we can see that there's no less immunogenicity, even relatively a little bit high compared with a little bit adolescents. So that's very good results. Our next speaker is Dr. Zhang. Zijie ZHANG is a principal investigator and a professor in the State Key Laboratory of Conservation and Utilization of Bioresources in Yunnan University. And he's also a member of professional committee of the Nucleic Acid Society of Chinese Society of Biochemistry and Molecular Biology. And he, in this conference, he will report the review on the immunogenicity. He will review on the immunogenicity and safety of phase 3 of the CoronaVac vaccine used in another special patients with chronic disease. So China has to, we do not have a high incidence in the country, so we cannot contribute to many of efficacy data. But we are doing extensively on the immunogenicity and safety data on different populations of our people. Dr. Zhang, please, the floor is yours.
Okay, thank you, Dr. Shao. Thank you for your kind introduction, and thanks for SINOVAC for the invitation. So I'll share my screen here. All right, so can you see the screen?
You can proceed, yeah.
Okay, great. So good evening, everyone. It's very honored to share our research about safety and immunogenicity of CoronaVac inactivated vaccine in people with 6 different types of chronic diseases at this meeting. So my name is Zijie from State Key Laboratory of Conservation and Utilization of Bioresource at Yunnan University. So currently, massive application of COVID-19 vaccines is a major approach to reduce SARS-CoV-2 transmission and COVID-19 symptoms worldwide, and thereby recover socioeconomic activities. An inactivated virus vaccine is one of the most widely administered vaccine type in China and in many countries, with over 2 billion doses vaccinated. However, many gaps of knowledge about application of the inactivated vaccine remains to be studied. For example, the application of inactivated vaccine in people with chronic diseases still lacks data support. Many studies have reported that COVID-19 patients with chronic diseases are more likely to develop severe symptoms and death risks. Immune protection against SARS-CoV-2 established by vaccine are particularly important for such special population with increased vulnerability. There has been several studies about people with underlying medical conditions on various types of vaccines. For CoronaVac inactivated vaccine, immunogenicity and safety in patients with autoimmune rheumatic diseases has been reported. In this study, we focus on immunogenicity and safety of CoronaVac in patients with 6 types of very common chronic diseases, including coronary artery diseases, chronic respiratory diseases, diabetes, obesity, cancer, and hypertension. Given that senior population are more susceptible to severe symptoms when infected, and chronic diseases are more common in senior people, our study mainly focused on people above 40 years old. So for 6 groups of patients and health controls, we aimed to evaluate the neutralizing antibody seroconversion rate 2 weeks after the 2nd dose of vaccination. We also collect and evaluate the adverse drug reactions occurred within 2 weeks after each dose. Additionally, we will compare geometric mean titer of neutralizing antibody across groups 2 weeks after the second immunization, as well as 3 and 6 months post-immunization. So this is an ongoing study, and tonight I'm going to share you the data of the first endpoint after the 2 weeks post-vaccination. We are currently still collecting the data for the 3 months and 6 months post-vaccination. And on the left figure shows the distribution of collected samples in 6 groups of diseases. Hypertension and diabetes patients were relatively abundant, while other 4 types of diseases were relatively fewer. Indeed, we are still actively recruiting additional participants to expand the sample size in these 4 groups. While these numbers are, well, currently this figure shows the number I will discussing today. So the age of our participants ranges from 40 to 91 years old. We divide them into a mid-age group from 40 to 59 years old and a senior group older than 60 years old. The distribution of age are quite close between diseases group and for both mid-age and senior participants. Another important variable that could significantly influence antibody response are interval between 2 doses. This variable is distributed in a tight range among mid-age and senior age groups. So to qualitatively analyze humoral immune response after the completion of 2-dose immunization protocol, we use neutralizing level of 8 as a cutoff and computed seropositive rate of health controls to be 88%. And combining all diseases group, 86 people exhibiting seropositive neutralizing antibody levels. And performing chi-square test on seroconversion rate between health controls and each chronic diseases groups revealed no statistically significant differences. So this indicates in terms of seroconversion, diseases people and health controls are equally effective. And next, we use neutralizing antibody titers measured using live virus test for quantitative analysis. So the geometrical mean titer of health controls are 30.4, and the geometrical mean of coronary artery diseases, chronic respiratory diseases, and diabetes are a little bit lower, but not statistically different. And other diseases are very close to health group. And we know that for diabetes, there are type 1 and type 2 diabetes, but they both are equally similar to the health control groups. And studies have shown age-related heterogeneity in immune response to COVID-19 vaccine. So we wonder if chronic disease could affect immune response to CoronaVac differently in mid-age and in senior participants. Indeed, we observe that in mid-age group, cancer and hypertension patients show statistically significant elevated antibody response. And in contrast, chronic respiratory disease and diabetes show significant lower antibody response in senior group older than 60 years old. This result suggests that chronic diseases could influence immune response in an age-dependent manner. However, we also note that antibody level differences observed in each age group are in relatively small magnitude and may not really contribute much to the clinical outcome. So next, we will switch gears from immunogenicity to safety evaluation concerning adverse drug reaction. So adverse reaction data were collected by survey filled 2 weeks after the second dose of vaccination at local clinics where serum samples were collected. So we surveyed occurrence and severity of 17 types of adverse reaction after each dose of vaccination. Local adverse reactions include pain, scleroma, swelling, flush, and rash, and itching. And systematic adverse reaction include acute allergy, skin and mucus abnormality, diarrhea, anorexia, vomit, nausea, muscle pain, headache, cough, fatigue, and fever. In our study, no severe adverse reaction has been reported. Most reported adverse reaction were level 1 with were level 1, and 6 of them were level 2 adverse reaction. So this figure shows the total count of adverse reaction in two separated doses. Injection site pain and fatigue are two most common adverse reactions, with more injection site pain after the second dose, as you can see here, and more fatigues are more common after the first dose. And in this figure, we further stratified adverse reaction events count by age and we further stratified adverse reaction event counts by age interval. So the trend that first dose caused more fatigue and the second dose led to more injection site pain are consistent across age intervals. So this is consistent across mid-age and senior group. For both vaccination doses, mid-age group reported more adverse reactions than senior groups. And looking at diseases group separately, we still find mid-age people exhibiting more adverse reaction than senior people. And thus, CNAROVAC is safe for senior people with or without studied 6 types of chronic diseases. Bad thing is that adverse reaction events are sparse in each diseases cohort and are not statistically powerful to compare across different diseases. But the good thing is that this suggests the vaccine is safe in general. So this figure shows the percent of participants in each group who have reported adverse reactions. The number of people reporting adverse reactions are also shown. And because total number of participants in each group are not equal, so the percentage is more comparable across groups. We can see in this data that percent of people reporting reactions, reporting adverse reactions are quite close, ranging from 14.6% to 18.8%. And it's basically very similar across different groups. We then use chi-square test to evaluate the differences between each diseases and health controls. So for combined chronic diseases patient and health controls, as you can see here, the chi-square are quite small and with a large p-value. So stratifying diseases type leads to similar result, as you can see here for six types of chronic diseases. So thus we conclude that adverse reactions to CoronaVac in patients with studied chronic diseases are not statistically different from that in health controls. We then stratified participants by age interval. So the bar plot shows that in mid-age group, larger fraction of people with chronic diseases reported adverse reactions compared to healthy controls. But none of those differences from control are statistically different. In senior people, disease groups and health controls have similar percentage of people reporting adverse reactions. And chi-square tests, of course, indicate none of diseases are statistically different from health controls. Thus, our safety data indicate that for senior group older than 60 years old, CoronaVac in people with studied chronic diseases are as safe as in health population. So that's all the data I'm going to share today. Let me summarize our result here. First, after two doses of immunization, seroconversion rate in people with 6 studied chronic diseases are not statistically different from healthy controls. Neutralizing antibody of diseases people are also comparable quantitatively to health controls, and thus, CoronaVac is as effective in people with chronic diseases as in healthy individuals. Overall, adverse reaction occurred in 14.6% to 18.8% of vaccinated people. Most adverse reactions are level 1, while a few level 2 occurred. Severe reactions were not observed in our study. So adverse reaction rate in people with chronic diseases are not statistically different from healthy people. Thus, inactivated SARS-CoV-2 vaccine, CoronaVac vaccine in people with 6 types of chronic diseases are as safe as in healthy individuals. So with that, I'd like to thank SINOVAC Biotech and Yunnan Professional Science and Technology Department for the support, and also our collaborator in Hainan CDC, as well as people in affiliate hospital of Yunnan University and my team. And I'd also like to thank all the volunteers participated in this study. Without their support, we'll not be able to collect all those data and do the study. With that, I'd like to thank you all for your attention. Thanks.
Thank you, Dr. Zhang, for your very good report. I mean, the people with chronic disease, especially in the seniors, are the major population or vaccine need to protect to reduce the mortality of COVID-19. To have a comprehensive profile, safety profiling, and also immunogenicity results for this 6 types of chronic disease people are very important. And we could all see all these, there are some differences in the immunogenicity, and that would help us to improve our current immunization strategy and to provide a better protection under the safety conditions for our seniors. Thank you very much. And I think no matter how much the immunogenicity safety data we collect in China, we cannot provide enough data to see the vaccine efficacy. And our colleagues in Chile, they have done great work for collecting this type of data of the protection data. But also now they are also moving to understand the immunogenicity and the safety data for their phase 3 trial. That is one of the most important trials for SINOVAC. So we will very honorably invite Dr. Alexis Kalergis, and he is a professor in the Faculty of Biology and Pontificia University of Católica in Chile. He is also associate investigator in the medical faculty of the university. Dr. Kalergis, please, the floor is yours.
Thank you very much, Dr. Shao, and I'd like to thank SINOVAC and Co-Benefit for the invitation to present our work. So I will share my screen now. Is it good? Can you see my slide?
Yes.
Great. Thank you so much. Good. So I like to just remind how pandemics have been affecting humans for many centuries, and the work that science does in a collaborative manner is important to develop vaccines and other approaches that could help to control the pandemic or the spread of infections. So that was the case for smallpox, a virus that was eradicated in the late 70s thanks to the work of science and public health worldwide. And that's been also the case for other diseases that are due to viral infections such as polio, measles, rubella, and mumps. So every time a vaccine is introduced in the population, there is a clear, when the vaccine is safe and effective, then you see this reduction in the numbers of cases in the population. So in the overall, vaccines are successful tools. They can lead to a decrease in the number of cases for several different types of diseases. And as long as the coverage of the vaccine goes up, the protection in the general population increases. So one of the things that we need to confront, particularly from Chile and other countries, is the gap between what we call industrialized countries and developing countries, where the number of vaccines and the access to vaccines and their specificity of vaccines might be different. So it's important that we try to fill this gap. And we published a paper in 2018 showing that those countries that produce vaccines have usually more successful vaccination campaigns or vaccination. The impact of vaccines is better in those countries that have their own manufacturing capacity. So in our region, places like Brazil, Argentina, Mexico, and Cuba are the only ones able of making vaccines. And Latin America is a huge part of the world in terms of the size of the region and the people. So in collaboration with SINOVAC, we from the Catholic University, we are trying to reestablish a capacity that Chile had in the late 90s, and that's saying to produce vaccines here. And these projects, we've been able to collaborate with SINOVAC in terms of just promoting, facilitating the proper landing of SINOVAC in Chile. And so far it's actually moving very well. Now, when confronting the pandemic with the need of a vaccine, we realized early after the pandemic was initiated that research and collaborations leading to vaccines for the SARS-CoV-2 are very important. And we initiated a study that published in Frontiers in Immunology in May 2020, showing that our design of a vaccine based on BCG might have a chance, because countries that use BCG as a vaccine for tuberculosis, they have a reduced rates of outbreaks as compared to those countries that do not have a BCG program. So we are currently still working on this vaccine. We think that the quality of BCG, of inducing a trained immunity response in humans, might be beneficial to prevent the inflammation that is one of the main causes of the severe cases of SARS-CoV-2 infection. So we think that our approach has some potential advantages that we are actually currently studying. So we been working all through the pandemic since January 2020 on trying moving forward with our a prototype that was designed by us at the Catholic University in Chile. These are the students and postdocs working on that. But at the same time, I mean, this work that we've done, which is just scientific work based in Chile, had led us to, was highlighted in important places like Nature, in this article, which I find very nice. You know, Latin American scientists joined the coronavirus vaccine race because no one's coming to rescue us. And also in New York Times, there's a coronavirus vaccine tracker where you know, our work's been mentioned. And of course, that led to, before the pandemic, we've been working on vaccines for a couple of decades up to now. And we initiated before the pandemic an interaction with scientists and collaborators in China in order to work on vaccines. And one of the main events was actually in, I think it was September, October 2019, as part of the International Congress of Immunology that took place in Beijing, where we had the chance to present our work and also learn of the work that SINOVAC was doing on vaccines. So we were planning to collaborate, and then the pandemic came, and SINOVAC developed advanced development of this inactivated virus vaccine that was published in Science. It's an approach that we appreciated from Chile because first, it's safe, it's known to be safe. Second, it could be used in all ages. Third, it has all the antigens from the virus. And finally, for a country like Chile, a vaccine that requires 2 to 8 degrees centigrade is easy to handle, and the country has experience dealing with such vaccines. So then we were, of course, in touch with, very high frequency, and we knew about the study that was published in Science, and of course, we knew about the clinical trials that were in progress, both in adults and elderly, that led to their publications in Lancet Infectious Diseases. And knowing this data in advance, of course, encouraged us to initiate a collaboration specifically on COVID-19, on top of what we were originally planning to do. And so we created a MOU and then several agreements that were supported also by the governments. And from our side, from the side of the university, together with SINOVAC, of course, we work on a clinical study protocol that was submitted to the ethical committees and the Chilean regulatory agency that is called ISP. That was approved in September 2020 and led to the registration of the trial in clinicaltrials.gov. This trial involved 8 different centers, different parts of the country, and has a central scientific lab that was doing all the immunogenicity analysis. Different places of Chile were participating in this trial. And in terms of safety, data were quite encouraging. So the vaccine leads to just to minor side effects. Most of them are pain at the site of injection, some headaches, but not really major side effects, such as we have non-anaphylactic responses, for example, as part of the trial. And we focused on a very well analyzed immune response, showing that the antibody titers become significant 2 weeks after the second dose, and that the second dose was really needed. And as we will see now, we're talking about a third dose or a booster dose. And equivalent results we obtained from the total people enrolled in the study, as well as elderly. And this is just the early data that was we submitted very quickly to medical archives, but it was accepted in Clinical Infectious Disease, a high impact journal. We did several tests to evaluate neutralization capacity of the antibodies. And again, we found that 2 weeks after second dose, a significant neutralizing capacity is observed in the subjects that were enrolled in the study. And this was, we were able to look and differentiate the immune response, of course, between adults and elderly. And this was very important data that were analyzed not only by the Chilean MOA, Ministry of Health, but also by other countries in the region. And we found that there was a significant increase in the neutralizing capacity of antibodies over time, both in adults and elderly. So the and this is just more data showing the same concept, different tests for we did direct neutralizing of the actual infectious virus, but also we used surrogate kits. We use a pseudovirus, and as I said, we had 4 different strategies for looking at neutralization capacity of antibodies induced by coronavac. But we also decided that it was very important to look at the immune response, at the cellular immune response triggered by the vaccine. So in collaboration with colleagues from La Jolla in the US, we got some peptides that specifically prime CD4 T cells or CD8 T cells from people vaccinated. And these are megapool peptides that can bind to MIC class 1 or MIC class 2. So we can differentially evaluate by ELISpot the presence of cells that recognize these peptides from the different antigens from coronavirus. And we could see that after the second dose of coronavac, there are significant T cells that could be detected in the circulation of the subjects enrolled in the study. Also, we had a paper that was just published showing that we have a few cases of COVID-19 as part of the study. We got a rate of about 2.5% of cases. So we analyzed very carefully the immune response of the people that are these breakthrough cases as part of the study, and found that the immune response after exposure to coronavirus led to a very strong activation of both antibody and T cell mediated immunity specific for the coronavirus, suggesting that the memory, the immune memory was there, and it was working in people that got infected with the virus. And these are the data, and we were able to compare between 2 different immunization schedules in terms of the response that subjects that were exposed and infected, they were just infected from the coronavirus circulating in the community, were actually responding properly in terms of this memory recall response after exposure to the virus. And we did the same with T cell responses, and now we are correlating the clinical outcome of these patients with their antibody and T cell levels against coronavirus. And we, as I said, we just published this paper a couple of months ago. So as part of this very virtuous collaboration, we have published now, I think so far, like 4 papers, and there are 2 of them under review. We have, as preliminary conclusions, that the vaccine is being well tolerated, and especially in the elderly. We have not severe adverse effects or withdrawals, no anaphylaxis from the study, and a very well characterized immune response after immunization. And we know that the antibodies are produced and can be detectable at high levels 2 weeks after second immunization, second dose, and also we are able to see and detect very clearly T lymphocytes that produce gamma interferon. Now, this has enhanced our capacities in Chile, so I think we're very we're confident that the capacities that we have built in terms of teamwork, protocols, lab protocols, equipment has actually improved our capacity to do research on vaccines. And of course, we've been working all along through SINOVAC, just connected to what was taking place in Brazil, Turkey, and Indonesia. Now, most importantly, as part of this collaboration, besides of the science and the clinical studies, it was the connection between China and Chile. We got very strong support from the government and a foundation in Chile that led to this clinical and scientific trial that allowed the country to access to doses required for conducting a mass vaccination program. So based on the data from all the trials that were going on CoronaVac, plus our data in the trial in Chile, the Chilean regulatory agency and the Ministry of Health approved the vaccine in January 2021, leading to the first shipments of doses for the use of the vaccine in Chile. And this is very important because this is probably the first time that we have in the history ongoing scientific research, clinical trials, and mass vaccination at the same time in a country. So the connection with between SINOVAC and the Chilean government led to this access to the doses required for mass vaccination, and we start seeing some of the benefits of that process during March and April. And it was very impressive that the way the Chilean government and just not only the government, but the local government and also the small healthcare systems that is really well prepared in Chile for vaccination led to a very efficient process of vaccination. And as you see here, most of the vaccines that have been used are from SINOVAC, and it's pretty amazing, I think, the way the vaccination campaign was able to control this major second wave that was taking place in the winter in Chile. Remember that we had winter during March and September. I mean, cold temperatures. The winter starts in July, but the low temperatures begin between March and April. And as you can see, the vaccination campaign was very contributed significantly to keeping down and controlling, reducing the number of cases of COVID-19 in the country. And this is thanks to the collaboration that we did with SINOVAC and the agreements that were signed between the university and SINOVAC and the government and SINOVAC. Now, the Chilean Ministry of Health did a effectiveness study that showed pretty clearly how effective the vaccine was after a couple of months of the vaccination campaign. So the lowest number is 67%. That's to prevent symptomatic COVID. But as you can see here, this effectiveness it's a very encouraging effectiveness, much higher, for example, than what you get with influenza vaccine, which is about 40%. Now, in terms of numbers of side effects out of the population studies, in July this year, there have been several reports, but this is one of them showing that CoronaVac is one of the vaccines that has the lower number of side effects. And this is, and it's been, if you look at this number, it's 5,000, but it's because most people got SINOVAC here as compared to other vaccines. And this is the number that really matters. It's about 36, about 100,000 doses that were administered. So this is pretty low as compared to the other vaccines that were used here in Chile. So the main conclusions, and this is what we really are proud of, is that first, we helped to contribute data about safety and immunogenicity of SINOVAC vaccine, and we were able to characterize the immune response and provide data for the proper use of the vaccine in the population. But the fact that safety, immunogenicity, and efficacy data from the controlled trials are predicting what's going on the population, as a scientist, to us, as scientists, for us, this is very motivating, very exciting. Now, some questions that we have already answered and the papers have been submitted is how long the immunity lasts, whether additional boosts are needed. We have already answered those questions. Capacity to neutralize variants. We also have that paper, I think was accepted. And also whether, and this is very important, the vaccine is safe, immunogenic, and efficacious in pediatric population. I just show you just a few data, and with this few slides are going to be ending. If you just look at the 014 or 028 schedule of vaccination, we were able to provide to the health authorities data showing that by 6 months after the first dose, there is a clear diminishment of antibody and T cells. That was sort of a sign for recommending that an additional booster dose might be needed. We were able to show that the antibodies induced by CoronaVac immunization lead to a cross-reaction with most of the variants of concerns. This is very important because we have no longer in Chile the original strain circulating. We have mostly Gamma and Delta. So having data supporting that the immunity induced by CoronaVac can neutralize those variants is important. And most importantly, we recently, and this is actually out already as a preprint published in medRxiv. I was actually posted yesterday. We were able to show a very significant increase of the antibody and T cell levels after a third dose of CoronaVac in people from the study, suggesting that in addition to the safety, the booster dose is very efficacious at increasing the levels of the neutralizing antibodies. This was 6 months after the first dose. As I said, this data are already posted as a preprint in medRxiv. So one thing that's very important, countries that have moved efficiently at vaccinating the population have confronted this problem, which is shown here in this slide. The virus is starts adapting to infecting people that have no immunity, and the pediatric population is of a special concern. And these data are from Israel, where you see that the frequency of pediatric population infected and sick because of COVID-19 is increasing because of the adult and elderly population vaccinated. And that's why we decided to move forward with a clinical study for pediatric population. We initiated the study quite a few weeks before the MOH decided to initiate a massive vaccination of children 6 years and above with CoronaVac. And these are the numbers for their vaccination campaign, which has been very active. And these are a little bit outdated, but these numbers are reaching now about 60%, 70%. So we are right now in the midst of this phase 3 trial that is multi-center with other countries, and this trial is going well. We already have the first safety and immunogenicity data that we share with the Chilean regulatory agency. They are particularly interested in the data between 3 and 5, which is the age group that is supposed to be next in terms of population vaccination. So this work was done combining basic research, clinical research, support from the public sector and the private sector. Of course, our collaboration with SINOVAC, international collaboration that goes across borders. And, but most importantly, I think, is the trust that citizens have on vaccines. And we have always used this approach, which is, I'd like to highlight this editorial from Nature showing that vaccine confidence requires radical transparency from science. And that's the way we've been handling our work on vaccine, as we did before for other studies we did on, for example, respiratory syncytial virus. So this is the core team working on the adult study, and I'm very thankful to them. They've been really a great team and work very hard. And of course, now for the pediatric study, which is larger, there are more people involved, and we got a very strong support from the university authorities, and most importantly, many scientists, clinicians, nurses have been critical for this work to be done successfully. So thank you very much. That's what I wanted to share today in this presentation.
You're mute, Professor Shao.
Thank you, Dr. Kalergis, for a very fruitful and comprehensive research results. And I think that what we see in China, the double harvest. You are not harvest only the public health interest to safeguarding your people, but also a lot of scientific achievement with publishing high-ranking papers and knowing a lot of unknown area of immunology with breakthrough infections, and that's very impressive. I think your visit to China in the autumn of 2019 is the right time for travel. Since then, our travel has limitations, and also I think the collaboration normally is the north source. I think from your work, we can see the south-south collaboration become more and more important, and also every research has to be supported by the infrastructure. We are very impressive by your the whole electronic database for your population, and everything goes very fast, and not only public health intervention, but also research at the same time. We should give a lot of remarks to your country and to your scientists. Thank you very much.
Thank you for your kind words.
Yeah, and I hope we can work together through cooperation and collaborations, and co-benefit our people and our scientific community, and then we can end this pandemic earlier. We can get rid of masks. I can see your picture, including our visit of Dr. Meng and a lot of Chinese colleagues. with a mask, and we could back to our normal life and a normal scientific collaboration without a mask. And I will end here. And thanks to all the speakers for the scientific session. Last but not least, I will invite Helen YANG, the General Manager for the SINOVAC Biotech, to give the final remarks. And Helen, please, the floor is yours.
Thank you, Dr. Shao. Your Excellency, distinguished guests, ladies and gentlemen, good day. Firstly, please allow me to express my sincere appreciation for your participation into the second co-benefit conference. I also want to thank all the speakers bringing us such informative presentations, and all the scientists, volunteers, and everyone behind all these studies. And SINOVAC aims to keep sharing scientific results to the public in order to support the vaccine application strategy. And so far, SINOVAC has achieved significant milestones through joint efforts from all stakeholders. Until now, we have supplied over 2.2 billion doses of CoronaVac to more than 50 countries. CoronaVac is currently become the most widely used COVID-19 vaccine from China, as well as in the world. SINOVAC become the Chinese manufacturer who export the most Chinese COVID vaccine to the rest of the world. And while the application of the vaccines, SINOVAC and the research institutions are continuously conducting additional research in order to fully study the products and its impact to the public health. We feel grateful to all the scientists who contribute to all this research, and you are all heroes to help the world return to how it shall be. As presented by all the speakers in the previous section, I think there are a great amount of safety data showing the vaccine is well tolerated among adults, elderlies, chronic disease patients, as well as children. And in terms of protecting against the variant, it also showed that a third dose booster of CoronaVac leads to a fast and durable recall of human immunity that shows good protection against a new variant of different kinds. In addition to research, we are also conducting joint vaccine production with different partners located in different continents in order to fulfill our commitment to make the vaccines more affordable and accessible in the world. Currently, we think the global COVID-19 pandemic is still a challenge. No one is safe until everyone is safe. Therefore, a global objective we are trying to meet is to establish a herd immunity, which requires a high level of vaccination coverage. So facing these challenges, we firstly need a vaccine which protects. As shown in the speaker's research, actually CoronaVac provides a strong effectiveness over hospitalized severe and fatal cases, and our booster shots plays even better. The protection will help reduce burden on our healthcare system in each country, and society can function normally, which is the primary objective of public health of using vaccines. But only protection is not enough. We also need to emphasize that the safety of the vaccine being used shall be regarded as a high priority, because this is an important factor to drive the increase in vaccination coverage rate, to allow a protective vaccine plays its role to improve public health condition. And this is very critical reason to behind why at the beginning, the Minister Sadikin from Indonesia said Indonesia's per day vaccination speed is the highest comparing to the other G20 countries, even for many of them have sufficient supply. And also Chile is a very good example of high coverage and preventing the pandemic after mainly using our vaccines. We feel grateful that we have the chance to work with countries like Chile and Indonesia and to make our contribution. Up to date for CoronaVac, the COVID-19 vaccine provided by SINOVAC, we have already achieved more than 1.8 billion doses that administered globally, and more than 100 million doses have been used in children group without any severe adverse reaction observed. And data shows CoronaVac is one of the safest COVID-19 vaccine in the world. And in view of this, more and more countries are now putting CoronaVac in pediatric use as low as 3 years old, including China, Cambodia, and Hong Kong. Some countries like South Africa can even lower the age limit to 6 months old to be vaccinated during the clinical studies. So in conclusion, COVID-19 outbreak is neither the first nor the last challenge to confront by humankind. Cooperation in vaccine scientific research and development should be an eternal winning strategy. And SINOVAC is ready to join hands with more countries and make unremitting efforts to provide more safe and effective vaccines to the world. As now the conference is coming to an end, and hope you all have already had an enjoyable and fruitful conference today. Thank you for listening, and I would like to take this opportunity to give my best wishes to all of you and all of the people living in your country. I'm looking forward to meeting all of you face to face, as we mentioned several times, by different people without masks, and we can shaking hands. With this, we conclude the conference today, and thank you for our host today as well. And good day, everyone. Thank you, and we're looking forward to seeing you next time. Thank you.
Thank you.
Thank you. Bye-bye.
Thank you. Bye-bye.
Thank you.
Thanks,
Bye.
Alex.
Thank you.
Thank you very much.